Hello everyone and welcome to the another video. Let's see what is the difference in calibration and validation in pharma company. We have heard this many times, so let's see in detail. So let's see our first detail. First is the definition. Calibration is a process or action that compares the measurement values of a measuring device or equipment against the reference standard and certifies the measurement accuracy. So this is very important to remember, measuring device equipment against the reference standard, right? So say suppose we have pH meter in our lab, or HPLC or balance. So while doing the calibration, and whatever the values we have observed, and whatever the values we have already got as a validated what you call that, validated equipments. Say suppose balance we are doing 1 gram will be validated, and if we observed 0.999, and what is the difference between 1 gram and 0.999, that will be our observed value, right? That will be our standard observed value. And if these values falls under our standard criteria or not, if it falls under our stated criteria, then it is calibrated, and if it is doesn't fall under our criteria, then it is fail, then it is out of calibration. So this is basically known as calibration. Validation is a detailed documented process of confirming that the equipment, process or system provides a consistent outcome. So validation basically we do for the processes like API or drug product or drug substances like tablets, capsule, whatever it is. And equipment also we do, few of the equipments we do the validation. And the system, actually this is for the process and system that provides a consistent outcome. Next is simple meaning. Well, what is does this means? It is a system performance checking. As you can see, pH meter, HPLC calibration. This is another reactor used in chemical synthesis or weight balance. So it is a system performance checking, and it is a performance analysis methodology. Like we do the validation of API, drug product, be it tablets, capsule, injections, syrup or ointment. So this tells about the performance analysis of a methodology, that our method is validated against the reference, against the reference or set documents. Next is features. What are the objectives? To nullify or remove the deviation by comparing with a reference or known standard. So you can see this is just an image where if we are using any instrument and we are going with the flow, we don't know whether it is correct or not. But if we are doing calibration, then we can easily know that what is the issue and where is the issue, and we can stop at that point only to get our wrong results. So therefore calibration is very important. Validation, to verify the consistency of predictable results of the method, process or procedure. As you already I have discussed, it is about the method, process or procedure. You can see process validation. The process validation goes on, and it tells us about the different issues or different right or wrong within the process. So this is mainly with the validation. Next is uses, to confirm that the equipment or measuring device is performing accurately. As I have already said, this is one of the equipment used in pharma industry. Means it is working perfectly. If it is not working perfectly, then we will get the wrong data and our interpretation will be wrong. Therefore it is very important to timely calibrate the instruments. Validation, to confirm the analytical method is performing accurately. This is just an example of linearity. We do by various methods like specificity, linearity, accuracy, repeatability. That is different thing. But to know that our method is performing correctly. Next is reference standard. It is done by comparing with a reference standard, as I have told in the first slide. And there are no standard or reference standards used, right? Like balance or pH meter or HPLC. There are certain set criterias, certain standards. So we have to know. So there are certain standards, and the calibration is compared against that. That okay, this is 1 gram or 1%, so it should fall within that limit. If it doesn't fall within that limit, then it is failed. So this is main difference in calibration and validation. Next is procedure. Executed according to calibration SOP. We have certain SOPs it is executed against to that, executed according to the validation protocol. We have validation protocol. For the validation we call it protocol, or we call it some STP, or we call it as a plan, we call also it as a plan. So if you want a detailed topic on this validation protocol, I will make a detailed topic. You can comment in the comment section. Benefits: to ensure specificity, linearity, accuracy, specificity. This thing is also done in the validation, but this is quite different in the calibration and validation, because we have a specific set of standards or reference standards in calibration. We don't have there in validation. This is just a parameter in the validation field. So what are the benefits? It will minimize reduction loss, reduction in utility cost, and then the help timely corrective action. If we are doing validation, at that moment only we will get to know whether it is right or wrong, right? Assure consistent production performance, ensure consistency of achievement of quality goals, reduce extensive finished product testing, right? We will reduce the costing and testing, ensure maintenance of the equipment, allow parametric result release, sort out the main risk of production liability, more rapid automation, right? This is one of the benefits of the validation. So that's all for this video. If you like, please like the video, comment if you have any questions, concerns, and do share with your friends, and subscribe the channel for more videos. Check out my other videos for more information. Thank you.